Hey guys, so we're going to be talking about chapter 9. So this is a relatively short chapter. So in this chapter we're going to discuss like the bacterial genome and how it's evolved and then different ways that it actually like replicates itself. So let's go ahead and get started. So there are multiple ways that a microbe can change its DNA. One way is just through spontaneous mutations, which is exactly what it sounds like. So whenever like it replicates itself, then there's an error. So that's just a spontaneous mutation. It happens all the time. And we constantly have mutations in our genome, but most of the time it's taken care of, so it's not a big deal. And then another way is called recombination, and there's 4 different types of recombination that we're actually going to talk about. So there's transformation, bacterial conjugation, so we've already kind of dabbled a little bit with the conjugation, and then there's transduction and transponsons. So we're going to start with transformation. So with transformation, this involves where the bacterium or just the pathogen in general will uptake DNA that is just free floating in the environment. It'll just uptake it, and then if it finds it advantageous and it's good for it, it'll start utilizing it. And then, or if it's not advantageous, then they can just get rid of it eventually. And there's 2 ways that transformation happens. There's natural and artificial. So natural is whenever it's just in the environment and the bacteria comes across it. And then artificial is something that we do in the lab if you want to make like an antibiotic resistant strain of some kind of bacteria. So there are 3 different ways to do artificial transformation. There's the electroporation, there's chemical, or there's heat shock. So with the electro one, what you do is that you have the bacterium, you have the free-floating DNA, and then what you do is that you give like electrical pulses to your culture, and then it will kind of shock the bacterium, and then it'll uptake it so the DNA can actually go across the membrane. And then there's chemically, so with chemical you use specifically calcium chloride, and the reason why you do a calcium chloride treatment to your culture is because calcium chloride increases the permeability of the cell membrane. So with the fact that the cell membrane is more permeable, meaning that it's more likely to uptake stuff, the free floating DNA can enter into the bacterium. And then there's heat shock. So heat shock is whenever you have your bacterium, you have the free floating DNA, and then what you do is that you literally heat shock it, meaning that you put it in like a hot water bath for, I think it's only like a few seconds if I remember correctly, and then you take it out. And with the heat shock, it literally shocks the bacteria, and then it just like opens up all of its channels, and then the DNA can free float in. So that's several ways to do it artificially, and then of course the natural way, which is where it just comes across it in the environment. It's just like, oh, you look cool, I'm gonna eat you. Okay, so this is specifically what I'm talking about in transformation. So what happens ideally is you'll have some kind of, it's called the competency factor, is synthesized and exported. So meaning that the bacteria actually made the CF factor and exported it outside of its cell. And then as the cell number rises, meaning that as we get more and more bacteria, the CF in the external, what it does is that it will activate a sensor, which is the kinase sensor on the cell surface. And then the fact that the sensor of the kinase transfers that signal into the bacteria, transcription will happen. And then since transcription is happening, there are some genes that are being produced and everything. And then with certain genes being produced, there's binding of extracellular DNA. So this is the mechanism of which transformation happens. So definitely know this mechanism. So like I said, CF, so that competency factor has to be secreted, has to be made and exported out of the cell. And then as cell numbers rise, you have increased CF levels, and with that CF will bind to this kinase. So this is the kinase right here. And then with the kinase being bound, what happens is that that will trigger transcription of the genome. And then with that, it will allow for binding of extracellular DNA. Okay, so that's transformation. Okay, so now we're going to move on to conjugation. So we've already kind of talked about conjugation a little bit. So conjugation is the transfer of DNA between bacteria cells, and there has to be cell contact. And that cell contact happens between the sex pilus. So I'm going to actually move to the next slide. So this slide shows the actual mechanism behind transformation. So what it is that you have this donor cell. So this donor cell has that extra genomic information, so that plasmid. So that's considered F positive. So the F factor is the plasmid, essentially, is the part of the genome that the other bacterium does not have. So you start off with the F positive cell, and then a sex pilus is formed between an F negative cell, so meaning it does not have the plasmid, as you can see. And then what will happen is that that sex pilus will be the bridge to which that plasmid will travel. So what will happen is that at the origin of replication will begin, and then once replication has begun, there will be a transfer of the F factor. So replication will occur, and then once that's occurring, then it will travel into the original F negative cell. And then what will happen is that once replication has occurred and all the transfer has happened, it will cease replication, and then they will be split. So the sex pilus will, and like the sex bridge will disappear. And then now, after all that has happened, we are now with two F positive cells. So you start off with an F negative and an F positive, and then you end with two F positives. So that's pretty much all I wanted to talk about. So know this mechanism of conjugation. So definitely know it. And then if you want, you can always go back on your own time and look at this animation of actual conjugation. Okay, so now we're going to talk about the next one, which is transduction. So this is the process of which a phage, so a phage is another form of external DNA, essentially. And what it does is that a phage is floating around, and it has something called a payload, meaning that it has DNA inside of it that what it's going to do is that it kind of looks like a tripod. So that phage will attach to the cell, like the bacterial cell, and then kind of like squirt in DNA. On the next slide, I'll show y'all. But what is happening is that phage is transferring host genome to bacterial. So, like I said, it like squirts it in. That's the best way I can describe it right now. And then there's two different types. There's generalized, and then there's specialized. So generalized is whenever it's bacterial genome only, and then specialized is whenever there's bacterial and viral. So that's pretty cool. But let me show you a diagram of what I'm talking about. So like this phage right here, so that's the phage. It kind of looks, to me, it looks like a lollipop. So what happens is that he will attach to a bacterial cell, and then once he attaches, that phage will kind of like squirt its genome into the bacterial cell, and then to make more of him. And then he'll make more of him, he'll replicate until there's so many phages that the cell will lyse, meaning that it will burst. So once that cell bursts, then there's a lot of phages just floating around. So it's pretty cool. And then now if it's the other mechanism, you still have the phage attaching to the bacterial cell, squirting in its genome and everything, but the difference is that the phage actually doesn't enter into the cell. In this case, what will happen is that it will detach. And then it'll detach and everything, and then it'll still make more of itself in this genome because it's squirted in its DNA. It's integrated its own DNA into the original DNA. And then the cell will eventually lyse, and then all those progeny, so all of the new phages, it'll go back with this new factor, with the new part of the DNA. It'll go back onto another cell, squirt in its DNA again, and then detach again, and then now it has two sites of the new gene. So in class, I'll be showing you these animations, but you can definitely always refer to them on your own time. They actually go a little bit more into detail as to what I'm doing. They're really good animations, so I highly recommend y'all looking at them. Okay. So, DNA restriction and modification. So, once a new piece of DNA has entered in the cell, what will happen? So, if new DNA is a form of a plasmid, so that means that it's capable of its own replication by itself, the plasmid will coexist with the other genomic information. So the new cell, it'll just keep replicating and everything. So it'll just make more of the cell with the original DNA and the new plasmid DNA. Now, if the foreign DNA is not able to replicate by itself, meaning that it needs aid, what will happen is a lot of times it will be degraded. Or it can actually get incorporated into the original genomic information. So, meaning that it will integrate itself into chromosomal DNA, and then from there it will be replicated, so on and so forth. So, it kind of just depends. A lot of this is very spontaneous. It's very, like I said, it's very spontaneous. It's kind of just up in the air for what will happen in the last scenario, whether or not it'll be degraded or not. It's kind of random. Okay, so this is an image of showing the phage DNA. So this is the phage. So that's why I said it kind of looks like a tripod. He's kind of cute. He's like a mutant spider. But what will happen is, like I said, that phage will squirt in or inject its DNA from the host where there is something called restriction endonuclease. So these are enzymes that cut up DNA. And then the host DNA is protected because of the endonucleases. So that's really nice. So restriction enzymes are specifically made by the bacteria to protect itself. So if for some reason like the bacterial cell did not have these restriction enzymes, then it's at risk for being taken advantage of, essentially. So that's good. And an example of a restriction enzyme is ECO-R1, so like ECO-R1. So that's a perfect example of a restriction modification system. So he's very effective in his job whenever it comes to like chopping up DNA. But these guys, they are very specific as to when they will chop up stuff. So they will look for a specific genetic sequence before they start cutting. So they don't just randomly go around cutting. They look for like a key factor to like signal them to do it. Okay, So, one way that they protect themselves, like the original bacterial genome, so as you see like right here, so this is like the phage DNA that got injected, and then you have restriction endonucleases being like, no, you're not good, I'm going to cut you. And then you have the bacterial genome over here that has these yellow dots on it. So these are what are called restriction sites, meaning that they're kind of like they're a protective measure. So this endonuclease doesn't actually start cutting up that genome. So that is how the original bacterial genome will protect itself, is by having these restriction sites. So that's pretty cool. Okay, now we have transponsons. So this is when genes can move from one chromosome to another, and that is called a transposable element or a transponson. So they exist pretty much everywhere in all life forms, but they're not like plasmids. These cannot replicate outside of a larger DNA molecule. So whereas plasmids, they can replicate autonomously, meaning by themselves, transponsons cannot. They have to have aid. And a lot of times these transponsons, they have certain enzymatic genes where they help in moving the element from one DNA molecule to another. So I'm actually going to show you a picture of that. So this is kind of like the simplest way to explain it. So essentially what is happening is that you have a insertion of a sequence that has a gene for the enzyme, so transpondase. So that's the enzyme. And then there are two restriction sites within that within that transpondase. And then what happens is that transpondase will bind to a specific region on the DNA. And then once it binds, what will happen is that it will essentially kind of be like cut out in a way, and then it will just reinsert itself into another place of the genome. That's the best I can explain it. I'm more concerned about like conjugation with y'all than I am about transponsons. Um, but essentially, what is happening is that these transponsons, they're hopping from one piece of DNA to another. They're being cut out, they're being cut and then copied and pasted somewhere else. So what is happening is that those enzymes that allow this to happen, those transpondase, what they do is that they generate duplication sites. So like these yellow sites, those are the duplication sites, in order for attaching to occur. So that's all I really want you to know. I just kind of just want you to know the definition of it. Okie dokie. So this is kind of like a big picture example of it happening. So let's say that we have a transpondin gene on a plasmid, and then all of a sudden, for some reason, whenever the plasmid and the chromosomal DNA become close, that transpondin is just like, I don't want to be on this plasmid anymore. I want to be on the actual bacterial genome. So what will happen is that reaction that I just showed you, where it will be cut out because of those enzymes and then reinserted somewhere else. So like I said, this is kind of just the big picture of it, where it can literally hop between genomes. Okay, so how does nature make these genomic changes? Like, how does that aid in evolution? So there are several ways that we can analyze genomes and how they like gain and lose genes. And then there's also you know what, actually, let's just scratch this. Never mind. Let's scratch that. But we are going to talk about natural selection. So we are going to talk about how do these populations, these species, how do they diverge? Meaning, how do they evolve and how do they become different over time? So, diverging means that you're starting off at one point, but then you start branching out. So, there's numerous ways that they do this. There's random mutation, which we talked about. So, this is pretty rare. Um, it doesn't happen very often. And then there's natural selection, which is Darwinism essentially, where if you have an advantageous gene, you will survive. So kind of survival of the fittest. You'll survive, and then the fact that you're surviving, you're able to make progeny. So you're able to make. So bacteria don't make offspring, but they make progeny. Um, and then with making progeny, then those progenies are able to replicate as well, and then the gene will live on until it needs to die out. And then there's reductive evolution. So this is when a trait is no longer needed for survival, and what will happen is that the gene specific for that trait that the bacterium have will eventually accumulate mutations, but it won't hurt the bacteria. So it's kind of just there. But the big one is natural selection. So that's the big guy. So that's how eventually organisms will diverge, and this includes us too. Like, this is kind of with any organism, it's not just bacterial, but with any organism. Living organism, let me clarify. Okay. So, we're gonna take kind of like a little sidestep. So, how do we identify microbes? So, we've already talked about like how do we identify them and everything. But sometimes it is very difficult because they are microscopic, of course. So that's why like we do stains, we grow them on specific plates and everything. So that's kind of the nice part of bacteria is that a lot of times you can manipulate their environment, and with that you can determine what they are and how do they act. So that's really nice. So we've already talked about how do we identify microbes. But specifically like practicality wise, so this is actually really important for your unknown guys. Um, so a common practice of how to kind of do process of elimination. So let's say that you start with an unknown and then you run all these tests in order to be like, this is him. So a schematic of that is something called a dichotomous key. So this is where you start off at one point and then you go to a next point. So I'm actually just going to show you the slide. So like you start off with doing like one reaction, and then from that you're going to determine like, okay, so if I did a gram stain, was it positive or negative? And based on that, your dichotomous key is going to evolve into some potential microbes. So this is very important for clinical practices because sometimes you don't know what your patient has. So you send it off to the lab, and the lab will do this for you, and they will do process of elimination based on certain, what's it called? Certain tests. So like either growing it or doing susceptibility tests, so like determining what's the best antibiotic. So this is another way of determining how to treat your patient. So that's the clinical setting. But then also for your unknowns, guys. So I'm just going to throw this in here. For your unknowns, this is actually what I want. Is essentially a dichotomous key or a flow chart of like what was your process of figuring out what was your bacteria. So definitely make a little mental note for your unknowns for this. Okay. So that concludes our lecture. So like I said, it was pretty short, guys. So we talked about the microbial genome differences. So like how do they, over time, how do they become different? So we talked about mutations, their evolution, transformation, conjugation, transduction, and transponsons. We also talked about DNA restriction, so that EcoR1 and how does it get modified. We talked about natural selection and the process of how do we identify a microbe, which was the dichotomous key. So that, like I said, that concludes our lecture. So I will see y'all in class.